Hi, lecture on quorum sensing and quorum quenching. What is quorum sensing and quorum quenching? Quorum sensing, it is the regulation of gene expression in response to fluctuation in cell population density. Quorum quenching, it refers to the mechanism by which bacterial communication can be interrupted. It is quorum sensing inhibition. Now let us look into the diagrammatic representation of quorum sensing. Now, quorum sensing, what is quorum? Quorum is nothing but a group of individuals, they sit together and decide an agenda and they implement it. Exactly here, the quorum sensing, the microbes, they are together and they decide upon the function, okay, a function that they have to implement. Okay, so that is what here the microbes do. Now in quorum sensing, the sensing bacteria, they produce and release chemical signal molecules called as autoinducers. Now here I've shown you the autoinducer that increase in concentration as a function of cell density. A single cell will not produce autoinducer, but that is not enough. When the population increases, okay, what happens? It may be the same species or different species in the same environment. Okay, when this concentration increases, only then the regulation of the gene expression takes place. Okay, so this concentration, increase in the concentration of the autoinducers is a function of cell density. That means there must be an increase in the population density. Then the cell to cell communication will take place. Okay, now here the microbes cannot speak, they cannot hear, but yet amazingly they communicate with each other. Communicated to carry out colony-wide functions. Now, what are the functions they carry out? Sporulation, bioluminescence, nitrogen fixation, virulence, conjugation, competence, and biofilm formation. Steps in quorum sensing. There are 4 steps in quorum sensing. First is the production of small biochemical signal molecules by the bacterial cell. Okay, they do produce within the cell these biochemical molecules. Now, they have to move out. How will they move out to the surrounding environment? Actively or passively, these signal molecules will be released into the surrounding environment. These signal molecules will be recognized by specific receptors once they exceed a threshold concentration. And there will be change in gene regulation. So these are the 4 steps in quorum sensing. Now autoinducers and its concentration. Here you can see that the extracellular concentration of autoinducer is less than the intracellular concentration of autoinducer. Okay. Now here the cell continues to replicate and they begin to produce autoinducers. So every cell that is produced or replicated, or every new cell will also begin to produce an autoinducer. Further, you can see that as the cell population increases, the extracellular concentration of the autoinducer will be equal to that of the intracellular concentration. Further, you will find that as the population density further or progressively increases, what happens? The extracellular concentration of autoinducer will be more than that of the intracellular concentration. So what happens here is the intracellular concentration of autoinducers stops moving out of the cell. There's no diffusion of autoinducers from within to that of the outside, basically because the extracellular concentration of the autoinducers is more. Okay, so this becomes absolutely or energetically inactive for the autoinducers within the cell. So this high concentration of autoinducers downregulates itself by inhibiting its own transcription factor, and the extracellular concentration of autoinducer is less than the intracellular concentration of the autoinducers. Automatically, when there is a downregulation by inhibiting its own transcription factor as far as pertaining to autoinducer, what happens? Automatically, extracellular concentration of AI becomes less than the intracellular concentration of autoinducer. That time, the current sensing stops a little bit. Now, current sensing in gram-positive and gram-negative bacteria. Now here, during reproduction, individual bacteria synthesizes autoinducers. I have shown you. Now here, this is gram-negative bacteria and here it is the gram-positive bacteria. In gram-negative bacteria, the autoinducers that is synthesized will move out passively, and they are acyl homoserine lactone, AHL, it's abbreviated as. And in gram-positive bacteria, this autoinducer is made up of peptide and it is actively transported using ATP binding cassette transporter system of the peptidoglycan layer in the cell wall of gram-positive bacteria. Whatsoever, whether it is gram-negative bacteria or it is gram-positive bacteria, the autoinducers, both of them, as they reproduce progressively, they will move out the autoinducers. Now, as the extracellular concentration of AI increases to a critical mass, the threshold makes it energetically unfavorable for the intracellular AI. Now, as the intracellular AI increases, this autoinducer binds to the receptor, triggering signaling cascade that alter the transcription activity and therefore gene expression. So see how important it is for a particular gene to be expressed and carry out a function. The quorum is very important. Quorum sensing is very important in these microbes, say gram-positive bacteria or a gram-negative bacteria. Now, the quorum sensing in gram-positive bacteria, autoinducer is a peptide. It is secreted by ABC transporter system, that is ATP binding cascade transporter. It is transported out. Example, Bacillus subtilis use quorum sensing to develop bacterial competence. Now here the mechanism is peptide signal binds to a sensor protein. The sensor protein is a histidine kinase. Now this kinase is located in the cell membrane of the bacterium. Histidine kinase becomes activated, and once this sensor protein becomes activated, this activation, it leads to the phosphorylation of the response regulatory genes and interacts with another regulatory protein which facilitates transcriptional activation. Obviously, RNA3. Okay, so this is the mechanism that takes place within the gram-positive bacteria when the peptide or the autoinducer is synthesized. Now, in gram-negative bacteria, the autoinducer, as I told you, is N-acylhomoserine lactones, and this is synthesized. This is nothing but a language of Lux I as well as R. Okay, it is synthesized by Lux I homolog and Lux R homolog. Now here you can see on activation how the AHL is synthesized. But what happens here? The autoinducer is synthesized and it is passively moving out of the cell. So extracellularly it is moving. But here what happens? The target gene, whatever gene this is, okay, whether it is sporulation or virulence or bioluminescence, or if it is from agricultural point of view also it is nodulation in a plant, okay, there is no transcription of this target gene because of the low density, that is population is very less, so there is no transcription. This Lux R is not activated. Okay, only when this binds to a particular receptor region of the target gene, then what happens is the target genes get expressed, and this happens only when the population density increases. So here you can see transcription is not activated at low density. But then as progressively the population density increases, AHL or the autoinducer concentration in the extracellular environment increases. Now here they come and they bind. As it increases, autoinducers binds to a particular receptor region on LuxR, and this particular complex will activate the target gene. As I told you, it can be any gene. Okay, it can be virulence too, sporulation, etc. So activation is activated at a high cell density. This is quite interesting. Suppose we want to control the virulence of this particular bacteria or any microbe for that matter, you just have to hold on control the quorum sensing. Okay, then you can definitely, if the gene is for virulence. Does plant response to QS signals? Yes. Response to QS signals. Plant response to QS signals, it is defense, primary metabolism, stress response, gene regulation, protein dynamics, cytoskeleton formation. Bacterial response to QS signals, virulence, biofilm formation, plasmid transfer, motility, stress response, protein folding, and EPS synthesis. Now, bacterial response to host mimic, whichever host they are infecting, the mimic, okay, exactly the almost the same as that of the autoinducer. What does the bacterial response are? It is virulence, motility, nutritional and stress response, plant nodulation, antimicrobial production, and adhesion. Now, current sensing, the power of microbes. Yes, because they can carry out many functions. And the key to unlocking the power of quorum sensing in soil is plant diversity. This is something pertaining to the environment itself. Now, I can just tell you that in case if there is no proper green cover, what happens is the population density of the microbes decreases. If the population density of the microbe decreases, it affects the soil, the structure of the soil, the nutrients in the soil, the water holding capacity in the soil will decrease. Now, when we consider a plant diversity, that means to say, I just will put across this way. If it is a monoculture, what happens? The quorum sensing will be very weak. But if there is plant diversity, the microbial diversity in the soil also increases. Okay, when the microbial diversity increases, it effectively functions more efficiently here. When the microbial diversity is more, current sensing is more, and it is more advantage to the physical structure of the soil, which will definitely support the plant, as well as it is almost like you have control over the climate change. These are the factors. See, when there is plant diversity, okay, you're planting different plant species, there is plant diversity. So your soil do have different microbes, that is a micro diversity you'll come across. There's quorum sensing. So when there is increased quorum sensing, what happens? The soil structure change. It becomes more nutrient holding capacity, more water holding capacity. So your water problem, because of all this, they are all interconnected. Your water problem is solved, environment pollution is solved, the soil structure is solved, the demand of the population for food is solved. See, everything, these are interconnected. It is in our hands. All right, so what is in our control? Just look into them and try to change that aspect. So then we can live on a eco-friendly, in an eco-friendly environment. Now, it enables multi-species crops and pastures to function more effectively than monocultures, produce more nutrient-dense food, and improve water retention in soil, making them more resilient and productive. See how powerful they are. If the soil microbiome is changed, it can switch on genes that were previously unknown. By detecting the biochemical signal, microbes can determine different plants growing in a particular soil. They determine how many functional groups are present. That is diversity in functional group for a single cell microbe to work together collectively and function like a multi-cell superorganism. Diverse ground cover is the most important factor in soil health. Plant diversity supports microbes that create a well-structured soil. Current sensing is at work when soil is sticking around the plant root rhizosheath. Okay, production of growth stimulating and plant protection hormones are present around this. So rhizosheath, as we all know, plays a very important for a plant. What are the advantages of quorum sensing? You can understand how bacteria communicate to each other. See, how powerful is this microbe? If I can explain to you here is a single DNA. Now, if you take the virus, okay, a single DNA, it just enters our body. They are benign. They will not. They are not very powerful. But once their concentration increases or population density increases or they replicate within our body, they can take over our body. That is the power of these microbes. So initially itself we have to see that this quorum sensing is inhibited when we consider the virulence side of the bacteria, fungus, or a virus. So powerful they are, you know. They can overtake us. That is what the entire body, they can hold it. Okay, so that is what is the power of these microbes. But you can stop the current sensing when, as far as the virulence is concerned, then yes, we are more powerful. You can understand how bacteria communicate to each other. Quorum sensing controls bacterial virulence. Therefore, QS has been considered as an attractive target for development of new remedial strategies. QS occurs within a single cell bacterial species and between diverse species. It enables bacteria to coordinate their behavior, especially during rapidly changing environmental conditions. By enabling QS to thrive, we can safeguard the world's agricultural resources. Like I did tell you in the previous slides, the power of the microbes. Now, that was about the quorum sensing. Now, we cannot go on with quorum sensing if it is virulence is concerned or biofilm formation is concerned. There are many disadvantages here. So now our savior is quorum quenching. It's abbreviated as QQ. It is a mechanism by which bacterial communication can be interrupted. It is done by inhibiting the production of autoinducers. It is achieved by degrading the signal molecules. It is required for inhibition of bacterial virulence in vitro and in vivo. The ability to disrupt QS may give one bacteria species an advantage over other, which depends on quorum sensing. Now, strategies for quorum sensing inhibition. There are three strategies. Now, here the signal generation you can inhibit. Okay, that is the signal precursor you can inhibit. Targeting the AHL signal dissemination. So the signal itself you can inhibit. Targeting the signal receptor. So the target gene, okay, the signal receptor complex that I showed you, right? So if you target the signal receptor, then you can inhibit the quorum sensing inhibition. Now the QQ in action, you can see here. Here, this is basically the inhibition of the synthesis of the signal molecule. So here you can inhibit the signal precursor. Okay, so when the signal precursor is inhibited, there is no synthesis of autoinducer. So they will not move out of the cell too. The next is you can go about with the degradation. If the autoinducers are in the external environment, you can go in for the degradation here, and this is can be of the enzymes that can degrade these autoinducers. And next is sequestration. That means you make them very inactive here or immobile here, and here you can also inhibit by interfering with the signal reception. So this is how you can stop the quorum sensing and via the quorum quenching. Here all the target gene then will be, obviously if the autoinducers are not there, the target gene will not be expressed. So some of the chemicals are halogenated furanones are structural mimics of QS signals. Enzymes such as AHL acylase, AHL lactonase, paraoxygenases degrade AHLs. Analogues that are synthetic are also developed to compete with the QS signals. Now, what are the uses of this quorum quenching signals? Prevention of biofilm formation, prevention of biofouling, biocontrol of plant pathogen, combats microbial resistance, decreases pathogenesis, virulence and competition. It serves as a lead in developing and formulating a new generation of antibiotics. Yes. These are few references I had gone. Keep your mindset positive.